Good morning, I'm Sarah. Today I'm going to be showing you a typical day for me at work. I'm a sperm researcher at the University of Sheffield, so I'm interested in male infertility. A male factor is responsible for up to half of the cases of infertility. And we're trying to do research to look at how sperm use energy to swim in order to be able to come up with new treatments and new options for couples that are struggling to try and conceive. And I'm about to head over from Manchester to Sheffield. So come with me and I'll show you what I do with my day. I'm doing an experiment today whereby I'm going to isolate the sperm out of seminal fluid and wash all the other parts of the seminal fluid away. And then I'm going to put the sperm cells in a tube in a magnetic resonance spectrometer, which should help us get a better idea about how sperm break down chemicals when swimming and keeping alive. I've speeded up parts of the video in order to show you more lab work in less time. Here I am layering a sample on top of a gradient which will later be centrifuged in order to separate the sperm from the seminal fluid. So I'm going to get a close-up of the sample after it's been centrifuged now. What you can see is that at the top there is a layer of seminal fluid. In the middle there's some poor sperm that we will get rid of, and at the bottom there's a very small pellet on this tube. Here I am disposing of the seminal fluid and poor sperm and just keeping the pellet of good sperm. I'm going to take this last little bit and resuspend it, mix it up in a fresh tube. I'm going to add extra PBS, partly to make sure that any sperm left in the bottom of the tube get rinsed out, and partly to make sure that any contaminants present get very much diluted, so that when we do our second centrifugation they get removed in the wash and we just get the sperm at the bottom. The second centrifuge is finished, and what I've done is resuspended the pellet with all the sperm in it in more PBS. So now all I have to do is put this sample into this tube and then we can start our NMR analysis. So we've prepared our sample and I've just got one thing to do before I run it, which is to look at how well the sperm are swimming in this sample. So to do this I'm going to use a CASA machine which is attached to a microscope so that we can see them. The CASA is telling me that this sample has about 57% sperm that are moving, and this is pretty decent for a sample. This is our NMR machine, and our samples are ready to go because this machine's got a big magnet in the cord, and it also means that the camera can't get too close. So I'm going to put the sample in, and then that will be run later on today. So I've now finished in the lab and I'm heading back into the office. The results are in from the sample we did earlier, so let's have a look. The spectrometer can detect chemicals in our sperm sample. These are identified as peaks. Two peaks present are choline, which is important in cell membranes, and lactate, which is produced when sugars are broken down to release energy. So thank you very much for being interested in how I spend my day. Hopefully you've learnt a bit about sperm research going on at the University of Sheffield. But now it's home time.